So now let's look at the Mendel's experiment. So for this, he took a tall plant and a short plant. So he took the uh, tall and short character as the uh, phenotype. I'll tell you what a phenotype when we go further in this lesson. So first of all, he took a tall plant. So the tall we use T, a tall plant and a short plant. Short is as short is the opposite of tall. We use simple T to um, to identify the phenotype of short plants. So normally in genes, when you take genes, we have two, the genome or the DNA, the nucleus, there are two genes for each character. So if it is tall, before going on to this, I'll explain it in words, right? So he took a tall plant and as well as a short plant. So he cross-pollinate the two plants. That means pollens of the tall plants were deposited on stigma of a short plants, and again the pollens of the short plants were deposited on the stigma of the tall plants. So that is between two plants, two types of plant. That means the short plant and the tall plant. Then he obtained the seeds and they were planted. So these, when the seeds got planted, uh, they gave uh, the plants that they gave were all tall, right? That means there were no short plants. So first he took a tall plant and a short plant. So he cross-pollinated them and got the seeds. Those seeds were planted and the plants uh, generated out of these seeds were all tall. And then after getting this, so we call these plants as the first F1 generation, right? That means first generation, F1 generation. So these plants of this F1 generation were allowed to self-pollinate. That means you had a tall plant and a short plant. So you cross-pollinate them and you got the seeds. These seeds were planted and the plants were all tall. You got all tall plants after germinating these, or after the germination of these seeds, they produced tall plants, not short. And again, he allowed each plant to self-pollinate. That means the pollens of this plant, the flowers of this plant were deposited on the stigma of the same flower, and within this plant they got pollinated, not between two plants, right? So again he got the seeds and they were planted. That means from a one tree he got the seeds. From each plant you have to get the seeds, and from one plant he observed that so once he planted, germinated these seeds, once these got seeds got germinated, he got a short plant and 3 tall plants. Right? So from each tree, you will get like this. So you can see the table down the page number 175. So rather than the tall short character, he has used the color of the flower and color of the seeds, shape of the seed, shape of the pod, color of the pod, position of flower, like those characteristics also he and he has taken all these characteristics as well. So from each test, so he has done the same thing for each plant. That means if you take the color of the flower as the character, he has cross pollinate a purple uh, uh, color flowering plant and a white color flowering plant, right? And then he has got the F1 generation, the plants in the F1 generation having only purple flowers, not white flowers. Again, each and every plant was allowed to self-pollinate. Then he has got around 705 purple flowering plants and 224 white color flowering plants. So when he get the ratio, he has got 3 to 1 ratio. That means for every when you get 4 plants, 3 will be purple color, 1 will be white color flowering plants. So he has observed that this pattern is happening for each and every character. And so he was so confused because why this short plants or the white color flowering plants didn't appear in the first generation, because if you see this table also, only one character is being seen in the first generation, F1 generation. The both colors are appearing only in the F2 generation. So he was trying to find find what is happening in the F1 generation, right? So he discovered a method, or discovered what is being happening in this procedure. So first of all, he named the feature in that means the characteristic. If you take tall plants, if the feature is tall, he has used T for the tall plants and simple T for the short, right? So once he got tall plants, that means when you cross for the cross pollination, he has to get a tall plant and a short plant. So for that pure breeding tall, that means I told you that 2 factors are involving in these determining these characters. That means I told you in the beginning, in the nucleus there are 2 For each and every character, there are two genes and involved. The two genes are involved. So for pure breeding tall plants, that means if you take a tall plant, pure tall plant, that means both its genes are capital T. That is pure breeding tall. Both of its genes are capital. Pure breeding tall. And pure breeding short means if it is going to be short, it is named as like this, right? There are some plants where some plants where they are and physically they can be seen as tall plants, but like this, it can be like this. No, one gene is from the I mean it's totally pure breeding. It can come from the totally pure breeding tall plants, and one the short the gene that is determined that is responsible for the shortness can be hidden, right? So for those we call recessive tall plants. No, tall plants with recessive short feature. Those are that means this Mendel found that because he found that these short plants do not appear in the first generation, so he identified and he discovered that this the gene that is responsible for this shortness is being recessive. That means it is being hidden in the first generation, but it's being presented in the next generation. That means F2 generation. So he built up three breeding types. That means pure breeding tall is like this, pure breeding short is like this, and tall plants with recessive short feature. But they are tall. They are tall, but they have a hidden short character, right? Character determining gene. So actually these we call as genotypes. That means these letters, right? Genotypes. And the things that you can be seen out, physically, right? So if you take this plant, it is tall. This is short and this is tall, right? So both these plants are tall, but this is short. So those we call phenotype, right? But there are three different genotypes, but there are only two different phenotypes, because genotype means how the genes are arranged. Here you have three different genotypes, and phenotypes always this is also tall, this is also tall, but this is short. So there are two phenotypes, right? So he discovered this. So he explained this. He explained what is happening in this first generation and second generation like this. So he got a tall plant and a short plant. The tall plant was a pure tall plant, and this was a pure breeding short plant. Right? They were called as parents. Right? in pollens, in the I think you all you'll learn this in the this reproduction lesson. These gametes or the sperms and ovum only have one type of genes. That means if you have the somatic cells, that means the cells in our skin, eyes have two different genes. But when it comes to the gametes, we call them as gametes, sperms and ovums. So pollens and ovum, we call them as gametes. So they have only one type of a gene, right? So gametes. And so in the first one gene, so what is happening? The cross pollination occurs. That means between the two plants. So you get the F1 generation. This is the F1 generation, and you get tall plants. All the plants are tall, right? And what is happening then? Then now these tall plants have this genotype, right? So I'll erase this part. So parents, F1 generation, right? Then in the second generation, what is happening? Self-pollination occurs within the plant, right? So the genotype of those parents will be like this. That means there can be two different gametes here. They will produce here also they produce, right? You can write the same thing, you will get the same thing like this, right? So TT and TP. So these are gametes. Right? Now what is happening? This gamete can combine with this gamete. So it will be TT. Look at carefully. This gamete can combine with this one. So that is TT. This one can combine with this one, TT. This one, right? So this is the F2 generation. So how many genotypes are there? That means how the genes are arranged. There are 1, 2, these two are same, 2, 3 types of genotypes, and how many phenotypes? Only 2. Because all these 3 are tall, this is short. That means because you cannot see the genotype, right? You can physically see the phenotype. So then you get 3 tall plants and 1 short plant. But when you go further and analyze the genotype, then you will realize that these although they we can see them as tall plants, when you go to the gene level, genetic level, you can see that these three plants have two different genotypes. That means out of these three plants, two will be having this genotype and the other will have this genotype. But they are tall plants physically. This is short. That's so he found this is the reason for this observation, right? So he found that this reason or the the gene determining the shortness is recessed. I mean, it is being hidden in the first generation, but it is being presented in the second generation. So that's the reason. So they will in the exam they will ask you to draw this actually, and you can use a um, Punnett square also. I'll draw the Punnett square too. You can use that one also to get these uh, generations, the genotypes and the phenotypes of these generations, because they allocate marks for these uh, genes, gametes, even these you will get marks. First one generation, parents, gametes, F2 generation. And for these also, they allocate marks. So this is very important. So you don't need to read the read this page number. Actually, is if you I mean, if you sometimes you may get confused if you look at the textbook, but if you can understand this, right, that means I'm not telling not to look at the textbook, but you can look at it. I am telling the page number is 170 576. No, you can, but don't get confused, right? So if you know how to do this, it's, I mean, they have just explained what is being happening uh, in these observations and these experiments. But you need to, the figure number 20.919 is very important. It is being asked in the exam. I mean, it has been asked in the past few years, and they have allocated a lot of marks for this. And I will show you now how to do the Punnett square. Right? So you have the same thing, you will put this into a square. So you have parents or P generation. Parents, that means parents generation. Parents or P generation. Better to write parents generation, right? So you have P, 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 right? Then gametes. So here we take only one gamete because both are same, right? Even though it's not, if you take like this, as I told you earlier, you will get the same thing, right? So it's no, so there is no need to get two different, I mean all the two. You can have one, right? So then you get the F1 generation when you cross pollinate. This is the F1 generation. So they will allocate marks for this, and this, right? So you can tell that all plants are tall. All the plants are tall. Then we come to the Punnett square, right? Here you have the female gametes and the um, male gametes. So this side you have the male gametes, female gametes, right? This side you have, right? That means in the previous case we took another plant like this. So this thing is being transferred to a square, right? So then you can cross this, combine these two, TT and TT, TT, TT. So this is tall, 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 short. That means 3 tall plants, 1 short. 3 to 1 ratio. But 3 different genotypes. 2 different phenotypes, 3 different genotypes. So again I'll write the, so this is very important, and then this is the F2 generation. You have to mark this as well. Although you have written this completely, if you don't have these things, you won't get marks, right? So be careful of those things. And I forgot to tell you that we are looking at only one character at once, right? Now we don't care about the flowering of these. We cared only about the short and the tall, short tall characteristic, right? But we didn't have the other characteristics, right? So because sometimes this tall plant can have the purple colored flowering plants, and the short plants can have white colored flowering plants, but we took only the tallness and the shortness, right? So that kind. So if you are taking uh, these purple color flowering plants and white color flowering plants, you don't need to care about the shortness and the tallness. But sometimes we have to we have to consider both characters, right? But at your level, we are not going to do those things. So if you take only one character for the test, if you are considering only one character, it is called as a monohybrid cross. That means only one characteristic is being tested. Okay, so a monohybrid cross. So it is in the page number 175. You can read that. So you can read 177, page number 177. So then, so it is, it's there to explain you this thing further. It's just a little exercise. And we'll then move on to basic concepts of genetics. So actually gene concepts about inheritance, so Mendel said that the features of an organism is determined by a special particular factors, right? So the identified particular factor is named as gene layer. So I told you this, so all our earlier days in these 19th century, they didn't have this name called genes. They have these characteristics. So, so he said that the features of an organism is determined by a special particular factors. So now today we call them as genes, right? So as I told you earlier, mm, if the two genes are similar for a given feature, that means so for this the two genes are similar, no? Capital T, capital T. This is simple T, simple T. For this feature, the two genes are same. So we call them as, so we call this feature as a homozygous feature, right? But if you take this TT, the two genes are different. We call them heterozygous. Homo means same. Homo means same. Hetero means different. Right? So earlier I told you about the phenotype and the genotype. Three different genotypes. The gene types are different. Right? There are three different gene types. Gene type is different, but normally although the gene type is different, the phenotype sometimes doesn't change. Like here, if you take this as a tall plant, this is also tall, but their gene types are different, two different gene types. But when you have same, the two genes are same. If the two genes are same, we call it as homozygous, right? So if the two genes are different to each other, it is called heterozygous. So homozygous means TT or TT. Heterozygous means, right? And gene expression also I explained about this phenotype and genotype. So gene expression is actually the combination of gene pair for a particular character is known as gene expression. So these are gene expressions, types of gene expressions, and within that you can have three different genotypes. So this is the gene expression, how the genes are expressed, either TT or simple T or simple T capital T. So this is so normally gene expression is described as the combination of a gene pair for a particular character. So phenotype, genotype I explained. So then we'll move on to nature of genetic material and genes. So scientists have identified the deoxyribonucleic acid. Normally they ask in the paper. DNA. DNA stands for deoxy, right? Ribo, deoxyribonucleic acid. DNA, right? Deoxyribonucleic acid. So this is the genetic material that transmits genetic or the features which are specifically or which are very specific to an organism from one generation to another generation. So you can remember, if you can remember, in the first lesson we talked about this DNA. It's formed as a double helix, right? Can you remember? It's formed as a double helix, right? You have these nitrogenous bases, right? They are adenine, thymine, guanine, cytosine, right? So this structure was discovered by two scientists called Watson and Crick. Watson and Crick in 1953, right? So according to the sequence of base pairs, I told you there are several base pairs in this DNA helixes, and according to the, they are arranged in a I mean they have been arranged according to a pattern. So that pattern determines the genetic material, genetic characteristics. So from my pattern is different to yours. That's why both of us are different to each other. So that's a little bit about the deoxyribonucleic acid, DNA.